My presentation is about fluorescent antibody technique. Immunofluorescence test. In 1994, Albert Kuhn showed that antibodies could be labeled with molecules that have the property of fluorescence. Fluorescent molecules absorb light of one wavelength and emit light of another wavelength. If antibody molecules are tagged with a fluorescent dye or fluorochrome, immune complexes containing these fluorescently labeled antibodies can be detected by colored light emission when excited by light of the appropriate wavelength. Here's the graph between excitation energy and wavelength. Fluorescent dyes used in immunofluorescent test. Fluorescein, rhodamine, phycoerythrin. Structure of fluorescein and rhodamine. Direct and indirect fluorescence test. Direct immunofluorescence test uses a single antibody that is conjugated directly to a fluorescent dye fluorescein. The antibody recognizes the target molecule, binds to it, and the conjugated fluorescent dye can be detected by the fluorescent microscope. Indirect immunofluorescence test uses two antibodies: a primary antibody which recognizes the target biomolecule and binds to it, and a secondary antibody conjugated to a fluorescent dye which recognizes and binds to the primary antibody and indirectly localizes the target for detection by the fluorescent microscope. Here's the diagram showing that direct immunofluorescent have only primary antibody and indirect have both primary and secondary. Fluorescent antibody test. The direct fluorescent antibody test DFA is used to detect the localized antigen in the patient. The tissue sample to be tested is treated with antibodies against the particular antigen that have been labeled with fluorescent dye. If the antigen is present in the tissues, the fluorescent labeled antibodies will bind, and their binding can be detected with a fluorescent microscope. Variation of tests are used to diagnose respiratory syncytial virus, herpes simplex 1 and 2, pneumocystis infections. Here's the mechanism of fluorescence observed and where antigen is located. Immunofluorescence. Qualitative, direct. Antibody to tissues. Antigen is labeled with a fluorochrome. Fluorochrome labeled antibody binds with uh, antigen present in tissue section. Direct immunofluorescence assay. Here violet shows tissue, orange shows protein, and sky blue shows primary antibody. Direct immunofluorescence. Mechanism of direct immunofluorescence. The indirect immunofluorescence antibody test IFA is used to detect pathogen specific antibodies in the patient. In this case, a laboratory generated sample of infected tissues is mixed with serum from a patient. A fluorescent dye labeled anti-immunoglobulin is then added. If binding of antibodies from the patient to the tissue sample occurs, then the fluorescent antibodies can be bound and fluorescence can be detected in the tissues by microscope. This technique is used to detect anti-nuclear antibodies, anti-double standard DNA antibodies, anti-thyroid antibodies, anti-glomerular basement membrane antibodies, and Epstein-Barr virus viral capsid antigen antibodies. Indirect immunofluorescence. Antibody to tissue antigen is unlabeled. Fluorochrome labeled antigen immunoglobulin is used to detect binding of the first antibody. Here is the color representation of mechanism of indirect immunofluorescence assay. Mechanism of binding of indirect immunofluorescence. Here's Trypanosoma pallidum from laboratory stock binds with specific antibodies in serum of patients. Antibodies binding to Trypanosoma pallidum plus fluorescent dye labeled anti-human immune serum globulin will react with any immunoglobulin. Gives the fluorescent spirochetes. Application of immunofluorescence. Immunofluorescence has been applied to identify a number of subpopulations of lymphocytes, notably the CD4, CD8, and T cell subpopulations. The technique is also suitable for identifying bacterial species, detecting antigen antibody complexes in autoimmune diseases, and detecting complement components in tissues. Localization of hormones and other cellular products stained in situ. Localization of antigen in tissue section or in subcellular compartments. Complex between antigen antibodies and anti-antibodies. Fluorescein labeled antibody attached to a Legionella bacilli. Thank you.